the macrophages. Now, the importance of macrophages from a physiological perspective has been recently highlighted by studies that show that macrophages colonize virtually all tissues. In several instances, they colonize tissues already as the tissue develops, and they perform a wide variety of functions that are really supporting the physiological properties of organs. So in this picture here, some of the diverse functional properties of macrophages are shown here, together with some of the general properties that these cells execute. So as you can see, these cells are scavenging cells. They facilitate maintenance of stem cell niches, they remodel matrix, they promote angiogenesis, or they regulate angiogenesis, they provide trophic support. So many of these functions of macrophages are actually not linked to their true or classical immunological properties, that is, immune stimulation or the capacity to act as local sentinels. So macrophages are actually much more than simple sentinels of the immune system. And one of the key notions that I think has arisen over the recent years is that the same physiological properties of macrophages are actually co-opted in a wide variety of diseases, such as cancer. And this really makes tumor-associated macrophages, for instance, a very important therapeutic target. Okay, so in order to understand how the biology of macrophages is controlled, I think we need first to try to dissect the various layers of regulation of this type of cell. So on the one hand, we should appreciate that the identity of macrophages matter. So some macrophages in some tissues originate already from embryonic precursors that colonize the developing organs and that persist virtually throughout adulthood in these organs. And this is the case, for instance, for the microglia or the brain macrophages. While on the other hand, other types of macrophages, such as those in the intestine, are subjected to continuous replenishment from a circulating monocyte. So this is likely to establish some differential behavior, which I don't think we have really understood yet. Then at some point, we have the capacity of macrophages to sense local signals and undergo local adaptation. And this is important for the capacity of these cells to exert their tissue-specific properties. And then as a final layer, we have the capacity of these cells to sense environmental changes and therefore mediate, for instance, responses to infectious agents or to tissue damage or other types of disease. So in a very conceptual point of view, we can put together these three layers of regulation. So identity, tissue specificity, and response to environments into a very simplified model, but I think it's very useful, model of genomic regulation of the macrophage, gene expression, and epigenomic regulatory program. So these are studies that have been carried out over the years by the lab of Gioacchino Natoli, by Chris Glass, by the group of Ido Amit and Ruslan Medzitov and many others. So these studies have actually helped us to define very fundamental principles of genome organization. So in particular, if we consider the same three layers of regulation we discussed before, we can basically assign to these three layers of regulation a very specific type of transcription factor. In particular here, the identity program in macrophages is executed by a limited number of transcription factors that we call lineage determining, and in particular in the macrophage system, P1 has been shown to be the most important one. We have tissue specificity, which is again driven by tissue-specific signals that activate transcription factors that mediate the local responses of macrophages. And we call this tissue-restricted transcription factor. And then finally, we have this third layer of environmental response, which is relayed upon the genome based on activation of stimulus-activated transcription factors, such as those of the NF-kappa B, STATs, of IRF family. So if you put together these three different types of transcription factors, and if you consider that all of these act in a combinatorial manner, So altogether, these transcription factors binds to thousands of sites in the genome of a macrophage, and therefore they dictate which genes will be expressed in a given time and in a given condition. So I think this is very important as a model, and this is not really just true for the macrophages, but it has been shown to function in the same way also for other types of mammalian cells, virtually all cell types that are capable of responding to the environment. Okay, so as you alluded at the beginning, the regulation of the enhancer repertoire as macrophages is actually highly dynamic, meaning that when macrophages are differentiated, they have a specific genomic organization here. And then when they get exposed to inflammatory factors, for instance, LPS in this case, so we see basically ChIP-seq tracks of some histone modifications like H3K4 monomethylation, which is one of the defining markers of enhancers. So we have different types of behaviors here. So these are the macrophages that are untreated, and these are macrophages that are treated with LPS for 4 or 24 hours. So we basically see enhancers that we call poised, which are already existing in unstimulated macrophages. And then these enhancers, when cells are stimulated with LPS, acquire this marker, which is H3K4/27 acetylation, which is a proxy of chromatin accessibility and transcriptional activity. So these enhancers are poised because they can become readily activated upon stimulation. We see opposite behavior, so some enhancers that are basally acetylated, so accessible, but which lose accessibility and histone acetylation upon treatment. And these are not repressed enhancers. And then there's this type of enhancer that we have termed latent enhancers. The group of Kritzglass termed them de novo enhancers, but they're basically the same. And you see that these enhancers are virtually undetectable, so they lack this monomethylation marker in unstimulated cells. But they acquire this defining property of enhancer just after stimulation. So we call this latent or de novo because they can be generated de novo after stimulation. Now, the cool thing about this type of enhancers is that once they are generated, they're never really erased over time. So they persist as long as the macrophages probably survives. And the fact that these new enhancers are generated following a first stimulation enables these enhancers and their regulated genes to be expressed in a faster way or in a stronger manner when cells are re-stimulated with the same or with a different type of stimulus. So these data were really important because I think they established a concept that now is very general and very familiar, and that is that in the immune system, even in the innate immune system, transient exposure to environmental signals not only drives transient behaviors, but also have persistent impact that condition how these cells will respond to further stimulation. So we can call these properties in many ways. We can call it short-term memory. We can term it innate immune memory. It doesn't really matter. The point here is that the macrophages are so plastic that they can sense what happens to them and then remember, at least for some time, compatible with their cell cycle state and their survival. And I think we are now all aware of the wide variety of implication and studies that have linked this capacity of macrophages to remember, okay, to states that have been defined in innate immune memory, that is the capacity of macrophages to respond in a faster way to multiple stimulation in response to infectious agents, or inflammatory memory, which doesn't really happen in the myeloid compartment, but also happen in the epithelial compartment or in stem cells. And I think another type of memory is the one that we observe in macrophages in tissues, whereby local exposure is also able to condition how these cells will respond to further stimulation. All right, so one of the long-standing interests, my personal interest and now of my lab is, as was said in the introduction, to understand how at the molecular level macrophage activation is controlled. And over the years, we have again investigated the inflammatory responses in macrophages and dendritic cells, first from a classical signaling point of view, and then as a postdoc, as it was said already in the beginning, from a genomic point of view. Now, the idea here is that the macrophages not only represents a very important biological system, but also a very nice model to study how inflammatory gene expression is controlled. So this heat map here is from a beautiful study from the Steve Smale group. Here you're seeing basically the expression, and these are the colors in the heat map of genes that are assessed by nascent RNA. So we're measuring here the RNA as soon as it is transcribed here. So you see how the different modules of genes, which are basically expressed and induced after stimulation with inflammatory factors with very specific kinetics. So this really highlights how precise and complex regulation of the genome organization and transcription factor activities basically shape the responses of macrophages. And so I'm also sure that we all are familiar with the concept of the macrophage activation spectrum. So the simple fact that macrophages can sense virtually any type of signal in their environment, and they respond to this differential stimuli, has led to the proposal of this model, whereby macrophage activation can be described as sort of a spectrum here. And in particular, at the end of this spectrum, these M1-like or M2-like states have been defined, M1 being basically corresponding to macrophages exposed to interferon gamma, so highly inflamed and activated cells, while M2 corresponding to the macrophages that are stimulated with IL-4. So this nomenclature, of course, has been really fundamental to understand the biology of macrophages, but with the development of novel technologies and novel experimental tools, this terminology is rapidly becoming, well, it is probably outdated. So from a conceptual point of view, I think that the way in which we can see macrophage activation is not really as a spectrum as highlighted here. So whereby a naive macrophages can acquire different type of behavior based on the type of signals that it is exposed to. Actually, if we think about macrophages, they always act within a context within tissues. So they are basically always exposed to multiple stimuli at the same time. So this concept here is that we term this signal integration model there, and that really builds about the idea that multiple signals must act at the same time to drive specific activation programs and functional programs of the macrophages. And I think we don't really know how these signals are integrated. Now, this is very important, I think, in virtually all cases of in vivo behaviors of macrophages. But I think this is really evident when we consider the extent and diversity of immune signals that are found in the tumor microenvironment, as highlighted here. So, and then we generally think about tumors as hot and cold, based on whether they have high levels of inflammatory responses and good responses to immunotherapy versus cold. Actually, what I think is that a relevant hallmark of the tumor microenvironment is that some degree of inflammation always coexists with some degree of immune suppression. So, for instance, if we consider very simply the danger molecules that are associated as tumor cells undergo necrosis, which activates the immune system. And these type of signals actually coexist with a variety of cytokines that instead suppress the activation of cytotoxic immunity. So I think this is very relevant conceptually, that we start to understand how this tug of war here is regulated. And I think we really lack mechanistic understanding of how this interaction is controlled. Okay, so we started to address this issue in the lab. And in particular, we started to focus on this molecule that is called prostaglandin E2. Now, PG2 is a lipid derivative of arachidonic acid. Is an eicosanoid. It's produced by via the activities of inducible enzymes such as the cyclooxygenase 2 and prostaglandin E synthase. And this is a lipid. It is basically produced by most, if not all, cell types. It is produced at basal levels, but the production of PG2 increases substantially during infection, during damage, or during cancer. Now, this pathway here is the same pathway that is targeted by COX-2 inhibitors or in general NSAIDs. So from a biological point of view, PG2 is a very interesting molecule. On the one hand, it's known to be an inflammatory factor. So PG2 induces vasodilation, induces fever, and that's why targeting COX-2 has anti-inflammatory effect. On the other hand, PG2 is also known to antagonize a different type of inflammatory responses. That is, it suppresses cytotoxic immunity. PG2 has been shown to suppress activation of CD8 T cells, of NK cells, and also to suppress the activation of macrophages in terms of their interferon responses. And here there's this other type of behavior, which I think is really interesting. PG2 also acts in stem cells. So it has critical importance in hematopoietic stem cells or in muscle stem cells or in the intestinal stem cells. So basically this molecule here acts as a global organizer of tissue and immune homeostasis. So lots of studies have been done on PG2, but I think the molecular mechanisms that control these different types of activities of PG2 are not really well understood. So we are investigating a lot in the lab into how PG2 controls these three types of behavior. And I think this has led us to identify some new mechanisms whereby immune modulation properties of PG2 are exerted, and this is mostly what I will talk about today. But we also identified how PG2 contributes to cancer-related inflammation, and we are working heavily into trying to see how PG2 controls tissue regeneration. But these are two aspects that I will not talk about today because it's too early and simply we don't have enough data yet. Okay, so going back to the point, our goal was to understand how PG2 acts as a molecular level in macrophages. So we took a reductionist approach. So we took bone marrow-derived macrophages from mouse and then stimulated these macrophages with LPS, which is a canonical activator of inflammatory gene expression. And then treated these macrophages with LPS alone or in the presence of prostaglandin E2 or other known immunomodulatory factors such as IL-10 or IL-4. But I will mostly talk about PG2 here in this condition. So this heat map shows the behavior of genes that are induced by LPS. So here the color represents the level of induction basically of each of these genes. So we see here that all these genes that are shown in the heat map are basically strongly induced by LPS, as you can see in this row over here, okay? Now, if we consider this other column, which is here, the LPS plus PG2 condition, we were able to define two groups of inflammatory genes. This group here that we call PG2 sensitive, you see are genes that are induced strongly by LPS, but are not induced as strongly or are not actually induced at all when PG2 is present and it is added to the stimulation mixture. On the other hand, these other types of gene, which we call resistant to PG2, are again equally induced by LPS, but no, PG2 has really no effect of these genes. So we first started to understand what were so having defined these very clear groups of inflammatory genes, now we could start to identify what were the underlying properties. And here, so basically these colors in the heat map reflect how much expression of any of these genes is dependent on the MYD88 adapter, TRIF, or IRF, basically signaling. So it doesn't really need computational to see that PG2 sensitive genes here are very highly enriched in genes that are dependent on IRF3, that are dependent on IFNAR signaling and that are actually induced by interferon alpha, while the resistant ones are actually very much depleted from interferon response genes. So these data indicate that different classes of inflammatory response genes are differentially sensitive to PG2-mediated suppression. And the genes that are antagonized by PG2 are enriched, are basically coinciding with those of the interferon program. Okay, from a mechanistic point of view, we could exclude that PG2 impacted on LPS-induced signal transduction or activation of transcription factors. But what we found was that PG2 acted very strongly and in a site-specific manner at the level of the chromatin. So this box plot here shows the result of an experiment, of a ChIP-seq experiments for H3K27, which, as I told you in the beginning, is a marker of active enhancers and promoters. Here we are focusing on enhancers. So these are basically enhancers that gain histone acetylation after LPS treatment. And then again, as we did before, we generated these two groups. In gray here, we call these enhancers PG2 resistant enhancers. So you see, these enhancers are actually gain histone acetylation here when macrophages are stimulated with LPS. And then here, PG2 really has no impact on the level of histone acetylation. Instead, these enhancers, which are now classified here in blue, are actually enhancers that gain histone acetylation upon LPS treatment, but these levels of histone acetylation are strongly reduced when PG2 is present. So that's why they are PG2 sensitive. So it shows us that there are thousands of enhancers that are induced by LPS but are negatively targeted by PG2. And you see here some of these examples. For instance, if you look here, the interferon beta-1 locus, you see here, this is the histone acetylation. These are untreated. You see how these enhancers, basically, they gain histone acetylation after treatment, but then these levels of histone acetylation are strongly reduced when PG2 is present. Okay, so what's special about these enhancers? So here I want to make a point here. So even when we look at the genomic organization of these enhancers in unstimulated macrophages, so in the absence of any type of treatment of these cells, we actually see that the PG2 sensitive enhancers have much lower levels of histone acetylation as compared to the resistant ones, which suggests that these enhancers are basally poorly permissive and closed. And this makes sense because these enhancers control the interferon response program, which has to be tightly controlled. Now, if we look at what PG2 does to these enhancers, you see that these very low levels of histone acetylation are actually further reduced when PG2 is present. And we basically see the same if we look at the levels of binding of P1 or accessibility by ataxic. So the idea here is that these PG2 sensitive enhancers, which are basally poorly permissive, are sensitive to PG2, and then PG2 further reduces the accessibility and histone acetylation, likely preventing this enhancer to be induced when cells are stimulated with LPS. Okay, so having defined these two groups of enhancers with very distinct genomic properties, we actually asked what was the difference in terms of sequence specificity and transcription factor regulation. So we perform motif enrichment analysis again here by comparing the enhancers negatively targeted by PG2 and the enhancers that instead are resistant to PG2. And so first of all, we find that these PG2 sensitive enhancers are enriched in IRF transcription factor, which is consistent with the fact that PG2 suppresses the interferon program. But I think the most relevant observation here was that when we look at PG2 sensitive enhancers, we find a very high enrichment of this motif that correspond to the MEF2 binding site. That was really interesting because the MEF2 transcription factors were never really studied in great detail at the molecular level in macrophages. We know that these type of transcription factors are extremely important for the development and function of cardiomyocytes, of neurons, or skeletal muscle cells. But there are evidence that they are important for adaptive immunity, but their role in inflammation were not really very well understood. So to investigate what MEF2 transcription factor do in macrophages, we generated cells that are deficient in either the MEF2A, MEF2C, and MEF2D isoforms, which are the ones that are expressed in macrophages, and then perform our usual set of genomic analysis. So we basically found that MEF2A is the key factor here. So what we're looking now is the behavior of histone acetylation in macrophages stimulated with LPS, in wild-type cells or MEF2A knockout cells. So again, here in blue, we are seeing the PG2 sensitive enhancers, and in gray, we are seeing the resistant enhancers that we have seen before. So what you see here is that the PG2 resistant enhancers are actually induced in a normal way, even in the knockout macrophages, which says that these enhancers are MEF2 independent. Instead, when we focus on the enhancers that are targeted by PG2, those same enhancers are actually very strongly dependent on the MEF2A transcription factor. Indeed, when we see the level of histone acetylation of these enhancers in MEF2A knockout macrophages, they are much lower as compared to the wild type. So this suggests that MEF2A is important for the regulation of these PG2 control genes. So we perform RNA sequencing analysis by comparing the behavior of wild-type macrophages and MEF2A knockout macrophages upon stimulation with LPS. And as you can see in this heat map here, we identified a whole set of inflammatory genes, as shown here, whose induction is really dependent on the MEF2A transcription factor. And these genes actually include genes of the interferon program. And again, to link the whole story with a PG2, if we look at the behavior of PG2 sensitive genes, we find that these PG2 sensitive genes are actually very highly enriched in the genes that are also MEF2A dependent, while the resistant ones are not. So the model here becomes that PG2 negatively targets a set of genes that is controlled by MEF2A, the MEF2A transcription factor, and that these genes are actually very highly enriched in genes of the interferon response. So this data also identify MEF2A as a key regulator of the interferon response in activated macrophages. Now, this is just not happening with LPS. Now here we are measuring interferon beta levels in wild-type or MEF2A knockout macrophages. So you see here, these are four different clones of MEF2A knockout cells. And you see here how these are the wild type here, and you can see how loss of MEF2A abolishes basically production of type 1 interferons by these cells in response to LPS. But we see basically the same results when we look at poly-IC or STING activation. So it's not just LPS, but all types of stimuli. And this also happens when we look at the expression of type 1 interferons in macrophages that are exposed to live pathogens. So this is established MEF2A is a central organizer or inflammatory response in the macrophages, and this is basically the model that we have identified. So here LPS activates in ways that we still don't fully understand MEF2A, and MEF2A is required to elicit expression of type 1 interferon genes and genes of the interferon response. And then PG2 probably in a cyclic AMP dependent manner is able to antagonize this process. And we are now really working very hard to understand how this mechanistically is performed. Then on a genomic point of view, our data also highlight that MEF2A is very important to maintain the basal accessibility of interferon response genes. So that loss of MEF2A elicit a sort of collapse of these enhancers. And then these enhancers become insensitive, basically, to further stimulation by LPS. So this study was published recently and was really the outstanding work of Francesco Cilenti, a former PhD student in the lab, together with Giulia Barbiera, a computational postdoc, and Marco Gemma as a senior postdoc who supervised this project. Okay, so I will now switch gears and present some unpublished study. Well, these are soon to be published studies where we actually try to start from an initial collaboration that we had with the group of Andres Hidalgo in Madrid and Lai Guangge in Singapore here. So these guys are true neutrophil experts here. Now, we have discussed macrophages as really being able to epitomize the capacity of cells to be plastic and to adapt to the environment. And I think these properties are due to the fact that these cells have high genomic plasticity, they have long half-life in tissues, and they actually are exposed to different types of tissues. Instead, when we generally consider neutrophils, these cells are considered to be poorly plastic because they express few RNAs, and these are not generally thought to be the most dynamically regulated cells, at least at the molecular level. These cells are typically short-lived. They live up to a few days in the blood, well, 1-2 days in the blood, few more in tissues, and they're mostly located in the blood. However, a series of studies in recent years have actually challenged this notion of neutrophils as really non-plastic cells and non-diverse cells. And in particular, these neutrophils here have been emerging as cells that are really able to sense environment and to acquire specific functional properties that are relevant in a wide variety of physiological and pathological states. So together with, again, Wang and Renato, we initially proposed that some sort of genomic regulation could underlie the capacity of neutrophils to be diverse and to perform their heterogeneous functions in vivo. And in particular, these predictions were actually validated by the group of Andres in this paper to which we collaborated, where basically he showed that by when analyzing the immunophenotype of mouse neutrophils that are isolated from the different tissues. So here in this UMAP, the colors represent macrophages from different types of tissues, and the colors are the different tissues. And as expected, so the macrophage here from the spleen or from the lung or from the brain, they have different immunophenotypes. If you do the same analysis for the neutrophils, again, you see that there's a cluster segregation. So neutrophils from different organs, they express different surface markers. And then the group of Andres also shown that this is translated into specific functions. And also looking at the transcriptome level, we see that there's these are actually the first single-cell RNA sequencing data that we generated in the lab. And as you can see here, this cluster here are the blood neutrophils. This one in blue is the lung and spleen neutrophils, and these other clusters here are the bone marrow and the spleen. So clearly you can see that different neutrophils from different tissues also have different transcriptional programs. So we were interested in understanding how this behavior would translate in the human setting. So we embarked in this project that is now coming to a conclusion. So we leverage on the availability of very, let's say, abundant and controlled cohorts of patients here at the San Raffaele Hospital. So we basically collected different cohorts of patients that were undergoing different types of inflammatory stresses, as generally defined as we could. So in particular, the type of patient cohorts that we could collect from this study was here healthy donors from which we collected peripheral blood and bone marrow samples. Now we have these very same donors that were exposed to GCSF. Now GCSF is a growth factor and differentiation factor for the neutrophils. It is used in vivo because it acts as a mobilizing agent for stem cells of the hematopoietic system, or HSCs. So these are donors of HSCs in procedures of HSC transplantation. So a different type of patient cohort that we investigated here are these HSCT patients. So these are patients with myeloid, hematological malignancies that undergo full myeloablative conditioning and then receive allogeneic stem cell transplantation. So we could actually follow this patient at a very short follow-up after HSCT, up to 2 to 3 weeks after transplantation. So monitoring the very early phases of immune reconstitution in these patients. And then second follow-up, which is typically when hematopoietic reconstitution is clinically defined as successful or not. And this as a longer follow-up as a progressive return to the steady state here. And then we have also these two other patient cohorts. These are patients with pancreatic cancer, advanced or metastatic. And we have also this other cohort of patients with IPMN that undergo surveillance or resection. And these IPMN are sort of neoplastic lesions that often, but not in all cases, progress to PDAC. So we collected basically blood and, where available, bone marrow samples from these type of individuals, and then perform a whole range of genomic and functional assays. So we'll briefly go through and try to highlight the key features that we have identified. All right, so the first thing we noticed, so typically neutrophils, when peripheral blood is analyzed and subjected to density gradient centrifugation, neutrophils tend to localize because of their high density in a high density fraction of a normal Fico gradient. However, in stress condition or during disease, some neutrophils with a lower density, and this is basically reflecting the fact that these cells are either immature or activated, so they have lost their expression of the granules. Some neutrophils can be located here in the mononuclear fraction, so together with monocytes. So these neutrophils are also referred to as low-density neutrophils. Okay, so by simply quantifying how much of these low-density neutrophils, if these low-density neutrophils are mobilized in the blood upon these type of stresses that I discussed before, so we basically analyze the presence of neutrophils, so defined as cells that express CD15 and CD66P. So these are neutrophil markers. So you see here in the peripheral blood mononuclear cell fraction of steady state individuals, neutrophils are barely detectable, but they become a very substantial proportion, to 50% of the PBMCs in GCSF treated donors. Now we see analogous results, of course, with quantitative differences when we consider patients that have stem cell that have received HSCT. So these are patients at the first or second follow-up or HSCT, and you can see that we can detect low-density neutrophils in the blood of these patients. And also we could see the same in patients with pancreatic cancer, but not really in patients from IPMN. So the conclusion here is that different types of stress, which are associated to systemic inflammation, elicit the mobilization and the release in the blood of neutrophils with a low density. But we don't really know what these low density neutrophils contain. So we actually asked, so what are the properties of these low density neutrophils, or LDNs? So a phenotype analysis of these cells revealed that these are population of cells that are actually highly heterogeneous and that basically comprise neutrophils that range from very immature neutrophils, even precursors of neutrophils, to terminally differentiated cells. So they're really a spectrum of cells that span the full differentiation program of granulocytes. And you can see very clearly here. So these are the expression of CD16 and CD11B. So these markers become progressively expressed as neutrophils differentiate. So in the blood of normal donors, we basically see 100% of neutrophils that express high levels of CD16 and CD11B. So these are the mature neutrophils. While if we consider GCSF treated donors, then we start to see the appearance of cells that express intermediate level or even very low to absent level of these markers. And we, by analogy with what we see in the bone marrow, we can call these neutrophils immature or neutrophil precursors. Now we see with some difference, we see heterogeneous behavior of these LDNs also in patients that receive stem cell transplantation or in patients with PDAC. Now, the observation that during this type of stress, there is the mobilization in the blood of cells that are phenotypically analogous to neutrophil precursors was very interesting. because it would support the idea that these cells are released from the blood and they may contribute to local differentiation of neutrophils, either in the circulation or even in tissues, although we don't have data for that. And in further supporting the notion that these CD16 low cells are actually neutrophil precursors, we analyzed the capacity of these cells to proliferate. So proliferation is a property, I would say a defining property of neutrophil precursors. And to this aim, we perform EDU incorporation experiments. So, these three groups here are neutrophil precursors, immature or mature neutrophils, as we define here. And then we, on the x-axis, we measure the frequency of cells that incorporate EDU in their DNA, which is a readout of how much these cells have proliferated. And then you see here that in cells from GCSF treated donors or HSCT patients, we see a substantial proportion of precursors that actually proliferate, while the immature and the mature neutrophils don't. So this type of stress elicits the release in the circulation of low-density neutrophils, which include neutrophil precursors and also terminally differentiated cells that together coexist. Now, we actually try to define what are the functional properties of these cells. So we focused on GCSF elicited neutrophils because here we had simply more cells to work with. And then we isolated low-density neutrophils, normal density neutrophils from control or GCSF-treated donors. And then we measure the capacity of these cells to produce reactive oxygen species, ROS, or to release neutrophil extracellular traps, or NETs. Now here, if you see these are two concentration of PMA. In this group here in gray, these are the normal density neutrophils from control donors, which readily produce ROS. Instead, if we look here at the GCSF elicited neutrophils, they produce much lower levels of ROS. And also, if we consider here the production of release of NETs, which we measure by the concentration of elastase in the medium, you see that normal density neutrophils efficiently undergo netosis upon stimulation with PMA, but this is not really the case when we look at the GCSF elicited neutrophils. So this suggests that these cells lack some terminal effector capacities. Instead, when we look at how these cells are capable of producing cytokines, we see that low-density neutrophils from GCSF-treated donors, when these cells are stimulated ex vivo with a toll-like receptor activator, they produce very high levels of cytokines, which are almost always higher than the ones that are released by normal density neutrophils from control individuals. And in most cases are even higher as compared to the normal density neutrophils from GCSF-treated donors. So the conclusion of this data is that the GCSF-treated neutrophils represent a heterogeneous population, which probably lack terminal effector capacity, in line with the fact that these cells are not fully and completely differentiated. Instead, these cells may have greater immune regulatory potential as compared to the terminally differentiated cells, because of their capacity to produce cytokines. Now, at the transcriptome level, normal density neutrophils, LDN neutrophils, and monocytes are really distinct. So these are bulk RNA sequencing of cells that we isolated from these different types of patients. What I want to stress here is that low density neutrophils express this module of gene here that is to some extent shared with the monocyte. And this gene module here basically contains genes that encode for regulators of cell cycle or protein synthesis of RNA metabolism, which again supports the notion that these cells can undergo and actually so molecular reprogramming and produce cytokines. LDNs also express variable levels of this gene module, which basically contains genes that encode for granules, which are progressively expressed during the neutrophil differentiation, in line with the idea that these LDNs contain basically non-differentiated neutrophils. And this is quantified in some way by this PCA. All right, so now the key point is we have defined that LDNs are highly heterogeneous, and they contain cells that are ranged from being very poorly differentiated to being terminally differentiated. So of course, to identify what are the properties of these cells, we needed to basically recur to single-cell RNA sequencing. So that's what we did. So we generated single-cell RNA sequencing data for more than 130,000 neutrophils, human neutrophils from blood and bone marrow samples of donors and the patients that we have discussed before. And the output here is this UMAP, from where you can clearly see now, of course, the number of clusters depends on where you set thresholds, but it's very clear that overall this transcriptional regulation of the neutrophils leads to a very clustered and diverse, basically, universe here. So we try to make sense of what drives this diversity of neutrophils, the human neutrophils in the blood. And the first key driver of diversity was identified in the developmental stage of the neutrophils. So if we superimpose the expression of genes that we know are expressed in neutrophil precursors, and these are these signatures here, so we see that these genes are expressed in this area here of the UMAP, which correspond to this cluster one. So we call these cluster neutrophil precursors. When we look at these other signatures of genes that are expressed later during the differentiation of neutrophils, we see this specific granule signature, which is expressed mostly in this group of neutrophils, which basically correspond to these clusters here. And then if we move on with genes that are normally expressed as neutrophil differentiate, now we are able to define basically a series of clusters that basically correspond to immature neutrophils or to mature terminally differentiated neutrophils. So to some extent here, by looking at this x-axis, it's not an x-axis, but the UMAP here, we clearly see that the differentiation program and differentiation state matters as per the neutrophil diversity. Now, on the other hand, so the impact of the type of stress is actually very much impacting on the diversity of the neutrophils. So if we look at here, now we are looking at where the cells from healthy donors from the peripheral blood or from the bone marrow distribute in the UMAP. You see here that basically the bone marrow and the blood cells here seem to define a sort of developmental trajectory that goes from here to here. Now, when we look at what's the differentiation trajectory of the neutrophils that we collect from patients, from donors that have received G-CSF treatment, you see here, you see how there's a major divergence from this initial trajectory. So this is basically where the G-CSF neutrophils differentiate. And if we look at neutrophils that are collected from patients that undergo stem cell transplantation, and this is particularly evident at very early time points after transplantation, we basically see another type of trajectory that goes like this. So it's very visually evident how the exposure to stress and the maturation state are the main drivers of this major diversity of the neutrophils. So of course, this can allow us to identify the genes that are differentially expressed at the end of these various different developmental trajectories. And I want to focus here on the HSCT condition, the stem cell transplantation condition, because we found that neutrophils from HSCT patients, they upregulate hundreds of genes, so a lot of them. And many of these genes belong to gene ontology categories that are related to interferon response. So this is very important because it may, on the one hand, support the notion that neutrophils are reacting to the chemotherapy or to the damage that has been done during the transplantation procedure. But we are actually working with the hypothesis that this interferon response signature may predict also how well the patients will reconstitute after transplantation. Instead, if we look at G-CSF, neutrophils from G-CSF treated donors, we actually see that different types of genes are upregulated, and mostly we see a mitochondrial and proliferative response. PDAC patients actually do not really show major changes in neutrophil gene expression. Okay, now the last piece of evidence, which I think is also very important conceptually here, is that this dynamic regulation of gene expression is actually very evident only when we consider the terminally differentiated neutrophils and not the less differentiated neutrophils. So here, the individual box plot reflect the level of expression of interferon response genes. Here in red, these are the precursor neutrophils, the early mature and immature neutrophils. So this is basically reflecting the differentiation program of neutrophils. And you see here how the interferon response are highly expressed in the mature terminally differentiated neutrophils, but not in, for instance, the precursors. Of course, comparing the HSCT patient as compared to the steady state. And you see here, when we look at the behavior of individual genes, you see how these interferon response genes are strongly upregulated in terminally differentiated neutrophils from HSCT patients, but progressively less so in less differentiated neutrophils. So the point here is that the capacity, we are concluding now that the capacity of neutrophils to respond to the environment increases as a function of their differentiation. So this is becoming now the model that we would wish to propose. So, upon exposure to environmental stresses, heterogeneous population of neutrophils are mobilized in the blood. So the blood at some point contains from neutrophil precursors to terminally differentiated cells. Now, these cells coexist in the blood and get exposed to the same circulating signals, but they don't react in the same way to these signals. So the precursors here of the neutrophils, basically, I would say they don't really undergo major transcriptional changes in response to this type of signals, while the differentiated neutrophils acquire stimulus-specific responses, namely interferon response during stem cell transplantation, proliferation and metabolism in GCSF, and a sort of general inflammatory response in PDAC patients. And I think this renewed model of how the differentiation of neutrophils is linked at the molecular level with their capacity to respond to the environment actually may provide an explanation of how the diversity of these cells can be acquired, and to some extent also support the notion that these cells might acquire longer lasting adaptation despite the fact that they are short-lived. Okay, so with this I finished. This is a study that is supposed to be coming out very soon, and it's really the work again from Elisa Montaldo, a senior postdoc in the lab, and Eleonora Rosito, a postdoc in the lab who is a computational scientist, together with former PhD student Valentina Bianchessi and another postdoc in the lab, Nicoletta Caroni. So with this I finished. And I would really like to thank the people in the lab who do this work. I mentioned them in the presentation. You see them now here very nicely dressed up because we are in the host institution. But this is how they look like when we go to conferences. So with this, I finished, and I will be happy to take your questions. And I thank you for your attention.
Renato, thank you so much for this really amazing talk. For people who are listening to the talk online, please type in your questions in the YouTube channel chat box. There's already a couple questions lined up. So the first question is, do you know if the mechanisms by which PGE2 limits expression of LPS target genes is conserved with other factors that also blocks LPS-induced gene expression, such as G-beta, or if those pathways are distinct?
So I would say the short answer is no. So we found that so we look at genes that were negatively targeted by IL-10 and IL-4. So some of the genes that were antagonized by IL-4 and IL-10 were shared with those that were antagonized by PGE2, but this occurred only for a subset of these genes. The dependency of MEF2A seems to be really specific for the PGE2. I don't think that IL-10 and or TGF-beta act on MEF2A simply because of the fact that we look at the enhances regulated and they are really distinct with different enrichment of transcription factor binding sites.
Cool. And to follow up on that, I have a couple questions. One is that it's really interesting you have this PGE2 resistant genes, and those genes based on their dependence on all the essential component downstream regulators for the TLR4 signaling, seems like they don't rely on any individual pathways specifically. Could you comment on why those genes are PGE2 resistant, and what's special about this group?
Right. So the key difference between the resistant genes and the antagonized genes is that the antagonized genes are basally poorly permissive. So they need chromatin remodeling to be induced. And I think that that's what makes these genes sensitive to PGE2, because PGE2 interferes with this process of chromatin remodeling. So the resistant ones instead are typically, they have higher levels of histone acetylation already in unstimulated cells. They are not typically accessible. They are typically expressed with very short kinetics. So these are early induced genes. So I think these are also the genes that don't really require chromatin remodeling to be induced. So I think the key concept now here is that the pre-existing genomic organization not only dictates the kinetics, but also how these genes are antagonized by suppressive factors. I have to say, so I didn't put here, but as we did this study, we actually find that we have this group of interferon genes which are negatively targeted by PGE2. We have a group of genes that are resistant, but we also identified a set of inflammatory response genes that are actually increased by PGE2. And I think this explains why PGE2 can have both immunomodulatory and inflammatory effects.
That's super cool. And going back to the point you mentioned with the MAF2A factors, and if you look at where MAF2A occupies in the enhancer and promoters between the PGE2 sensitive versus the repressive groups, do you see the binding of MAP2A are different, or potentially they may require additional signal or regulators to distinguish these two groups?
Yeah. So the most evident difference was that the PGE2 sensitive enhancers were basally, so in unstimulated cells, they were occupied by higher levels of MAF2A as compared to the resistant enhancers. So what we saw is that, so the idea is that MAF2A controls basally these enhancers. Indeed, when we remove MAF2A with a knockout, these enhancers tend to lose their basal histone acetylation. In other words, removing MAF2A recapitulated the effect of administering PGE2. So the idea is that MAF2A is controlling the basal organization and therefore enabling these enhancers to be activated by stimulation.
I see. And there's one more question from the chat box on YouTube. So do you know if the proliferation of neutrophils precursors modifies their capacity to progress towards their more mature phenotypes?
No, I don't know that, but we know that the other way is definitely true. So having these continuum data sets of single cell RNA-seq of developing neutrophils, now it becomes really possible to compare the gene expression programs of neutrophils at analogous differentiation stages, which was something that couldn't be done by using classical and discrete markers. Now, what we saw was actually even neutrophils at the same differentiation stage, they basically modulated the dynamics of expression of granular poiesis genes. So in other words, neutrophils from GCSF treated donors tended to express either prematurely or persistently genes that were associated with a particular stage of differentiation. So the model now becomes that exposure to stress is impacting probably the speed or the dynamics of differentiation. I'm not sure of the other way around, and especially of proliferation. We haven't tested that, but that's a super important point.
And a follow-up on that between the precursor immature neutrophils and mature neutrophils, do you think they have different capability of sensing the tissue microenvironment? Of sense,
yes. Of sense, yeah. So we in the study actually we focus on the interferon response. So we were pretty amazed by the fact that in HSCT patients where you have elevated levels of interferons, the mature neutrophils upregulate interferon response genes, but the immature ones don't. So to model this in a, let's say, in a steady state condition, So we took neutrophils from cord blood. They basically include, again, immature cells and mature cells. So we stimulated the full set of genes ex vivo with interferons, type 1 or type 2, and then did single-cell RNA sequencing. So what we found is that even in that case, the response to interferon given as a signature was typically happening in the most differentiated cells, but not in the less differentiated cells. And this could be, this would sound trivial, but one thing that we could explain this behavior was that the expression of the receptor increased during neutrophil differentiation. So in this case, the explanation was pretty simple, no? So we have neutrophils which are committed but don't express the receptors for the type 1 interferon, so they don't sense or respond so heavily to interference in the circulation. But as soon as they differentiate, they upregulate the receptor and they become sensitive. So a speculation could be that this expression of receptor is also linked to progressive regulation of the epigenome, which would make the differentiated neutrophils more primed to regulate genes in a highly dynamic manner as compared to the less differentiated cells. But that's a speculation for the moment.
Cool. Thank you so much, Renato. So if there's no more question, thank you again for today's wonderful seminar, Drawing Early Career Immunology, and hope to see everyone in 2 more weeks. All right, I think we're back here.